Thank you. So hi everyone, my name's Addie Woicik, and it's a pleasure to be here today talking about our work Pisces, where we use a cross-modal contrastive learning approach for drug combination synergy prediction. This work is led by Jiacheng Lin and Hanwen Xu. So today I'll be talking about synergistic drug combinations, where the therapeutic effect of applying two drugs together is more than additive compared to applying them separately. And these combinations are really valuable clinically, because they can lead to better treatment results with fewer side effects, improving patient outcomes overall. For example, the drug ramucirumab taken in combination with erlotinib was shown to extend progression-free survival times in non-small cell lung cancer patients and was approved by the FDA in 2020. In addition to cancers, drug combination therapies have often been proven effective to treat other complex diseases, including HIV and Alzheimer's disease. Now, even though these combinations are so valuable clinically, they're also extremely rare, which makes them expensive and just otherwise challenging to identify, even in large-scale therapeutic screens. To give you a better sense of the scale of this challenge, I want to briefly discuss the dataset we use in our work called GDSC Combo, which was published last year. Now, this is a fabulous large-scale therapeutic screen for drug combinations in cancer, and they include 125 cancer cell lines across breast, colorectal, and pancreatic cancers, and test slightly over 2,000 different drug combinations. In total, the dataset contains slightly over 100,000 triplets, where a triplet contains two drugs and the cell line they're applied to, and each triplet is then associated with a binary synergy label. So the first thing I want to point out here is that only about 20% of the possible triplets are screened, where here I've already restricted possible to be the drugs and cell lines that are measured at least once in the dataset. Then of the triplets that are measured, only about 5% of them are synergistic. So this figure from the GDSC combo paper shows the properties of drug combinations in breast cell lines, where red indicates synergistic activity. So from this, we can note that first, the experimental space of possible drug pairs and cell line disease models is extremely large, and even these large-scale therapeutic screens can only sparsely measure this space. Then also, these synergy labels are highly imbalanced, and the positive synergy class that we're so interested in is very rare. So what we and others have proposed to do to mitigate this challenge is to use machine learning to predict whether or not two drugs will be synergistic in a given cell line. We can then use these in silico results to prioritize experiments for validation in therapeutic screens. Moving to a machine learning problem setting then, we consider an input as a treatment triplet containing two drugs and a cell line, and our goal is to predict whether or not they'll be synergistic. So a deep learning model tackling this problem can either explicitly or implicitly learn representations for the drugs and the cell line and use these representations to perform binary classification. Now, any model following this pretty general framework has several important decisions to make, and one of which is how should we best embed these representations and how should we combine them to learn something that tells us useful information about the treatment. And for what I'll be focusing on for most of today, we can also ask, how should we best represent the drugs? And this isn't a trivial question, because even thinking about how we should give the drugs as input to the model has multiple decisions that we could make. These include the SMILES sequence and the molecular graph. So as an example, for the drug temozolomide, the SMILES sequence looks like this. It's basically a 1D string telling us about the atoms in the compound and how they're connected to each other. And the molecular graph is a 2D graphical representation containing similar information. Now, both of these modalities have advantages and disadvantages. So starting with the SMILES sequence, because this is a 1D sequential input, we can directly leverage powerful techniques from natural language processing to easily produce embeddings. However, some previous work has shown that SMILES-based representations can struggle for more complex drugs, and in particular for drugs that contain many ring structures. On the other hand, the molecular graph can more precisely characterize some of the features of the compounds, for instance, by using edge weights to indicate things like bond distance. But it can be difficult to embed graphs with a large diameter. So we can see here that there exists some trade-offs with respect to which modalities might best represent different drugs. Then in 2021, Xu et al. Found that the SMILE sequence and molecular graph for a given drug are actually complementary. This means that if we can learn drug embeddings that integrate information from the SMILE sequence and the molecular graph, we can improve the representational power of our learned embedding space. And one way to try to perform this integration is with contrastive learning. The intuition that we can have here is that if we have any given drug, but multiple modalities to represent the same drug, embeddings based off of those modalities should be similar. And in order to kind of define what similar means, we can add that embeddings of different drugs should be different. And this connects very nicely to the field of contrastive learning, some of which we've gotten to hear about this weekend. But just to make sure that we're all on the same page, I'll briefly walk through an analogy with images. Maybe we're interested in thinking about shapes, and we think that a triangle is still a triangle, irrespective of the way in which it's rotated, and these are just different views of the same shape. At the same time, a triangle is not the same thing as a circle, and it's not the same thing as a square. Then let's imagine we have a model that is trying to learn how to best embed different shapes. In a contrastive learning framework, we'd say that these triangles form what are called positive pairs with each other, so we want their embeddings to be close together in our learned space. Different combinations of shapes then form what are called negative pairs, and we want their embeddings to be far apart. So this translates pretty directly to our drug modality integration problem, where we can say that the molecular graph and SMILE sequence of the same drug are just different views of that drug, so form positive pairs in our contrastive learning framework and should have similar representations. Then combinations of different drugs are, of course, different and should have distal representations. The key idea behind Pisces is to extend this basic contrastive learning framework to multiple inputs. So continuing with our shape example, maybe now we're taking as input pairs of shapes, perhaps to see whether they fit together, and we still think that rotation doesn't matter. But now our inputs are tuples containing two shapes that can be rotated independently from each other, but as long as I'm comparing two inputs that are still the same pair of shapes, they form positive pairs and should have similar embeddings. And again, input tuples containing different sets of shapes are negative pairs and should have different embeddings. So now we can move back to think about our drug synergy prediction problem and note that for a treatment combination that contains two drugs, the modalities that we are using to represent those drugs aren't impacting the treatment, and they aren't impacting its synergistic properties. Then, using our contrastive learning framework, we can say that all embeddings that we're learning for that same treatment triplet should be similar, irrespective of the inputs, or input modalities. So now we can look at the Pisces model as an example. So taking in two drugs and a cell line. For the drugs, we consider both the molecular graphs and the SMILE sequence as inputs. We embed the molecular graphs using a graph convolutional neural network and the SMILE sequence using a transformer, producing 4 different embeddings for those 2 input drugs. We then also embed the cell line using the overexpressed gene set in the protein-protein interaction network. Once we have these embeddings for the different drugs in the cell line, we want to integrate them to learn something about the treatment as a whole. And the way we do this is we first take an embedding representing the first drug, then concatenate onto that an embedding representing the second drug, and finally add in the cell line embedding. So now we have these 4 different treatment level embeddings that are representing that input triplet. We then have a multi-layer perceptron model that can take these embeddings and output our synergy prediction. But because these 4 embeddings are representing that same input triplet, we treat these as our positive pairs in the contrastive learning framework. We can then train Pisces end-to-end with a combination of 3 objectives. So our first objective is based off of the quality of our synergy prediction, because obviously we want to perform the task well. We also have a term based off of the contrastive performance, so whether the treatment embeddings for the same treatment triplet are similar and different for different treatments. And finally, we include a modality agreement term, where we want the synergy label that we predict based off of the SMILE sequence to be the same as what we would predict based off of the molecular graph. Now that we've gone through the model, we can start looking at some of our results on the GDSC combo dataset that I talked about earlier. In order to understand the value of this modality integration, we compare against several existing methods. The first three are deep synergy, ODNN synergy, and pro-deep-syn, which we can think of as being loosely based off of the SMILE sequence. They're really taking as input things like the molecular fingerprint and drug descriptors, which can be computed directly from the SMILE sequence and are also sequential inputs. We also compare with graph synergy, which uses the drug's target genes in the protein-protein interaction network, and deep DDS, which uses the drug's molecular graph. And then as a reminder, Pisces is going to be a multimodal model where we take both the SMILE sequence and the molecular graph. We compare the models in terms of their balanced accuracy, AUPRC, F1 score, and Cohen's kappa, so for all of these, higher is better. And first, looking at a standard 5-fold cross-validation setting, we see that Pisces, which is the dark green bar on the far right, is outperforming the other methods. We next considered a slightly more challenging setting where we stratified our test set according to the drug pair. So in this case, triplets in the test data are for a combination of drugs that we've never seen together before during training. And in this case, we see that Pisces still outperforms the other models, and actually this relative improvement is larger than it was in the easier cross-validation setting. We think this really speaks to the generalization power of performing this modality integration step. Finally, we also considered a test setting where we stratify our results according to the cell line, so now trying to predict for cell lines that we've never seen before. And this is definitely a more challenging setting, but it's also really valuable because it best reflects what we might like to do clinically, where a new patient might come in and we'd like to understand what therapy might work best for them. And what we see here is that performance of all of the models degrades, but Pisces is still performing the best on average, which is encouraging. We next sought to analyze how Pisces might be able to better generalize to test data. So here we're looking at the embedding spaces of Pisces as well as the two graph-based methods, which were the best performing comparison approaches. And so looking at the test triplets, we don't see a particularly clear clustering behavior for either deep DDS or graph synergy with respect to the synergy labels. If we look at Pisces, though, we see much more clear separation with respect to synergistic properties. We think this is pointing to the fact that Pisces has learned an embedding space that's well organized according to synergistic properties, and that by performing this modality integration at the treatment level, we're able to generalize to the test data as well. Now, in addition to looking at kind of test splits as a whole, we did a further analysis where we considered how individual drug combinations were performing. And what we found here was that Pisces had greatest improvement relative to the other methods when one drug in the combination had many ring structures and the other drug had a large molecular graph diameter. So as an example, we had an over 200% improvement in AUPRC for the combination of vinylrelbine and lincitinib, where vinylrelbine is shown on the top here, and we can see that it has many ring structures, and lincitinib has a molecular graph diameter of 31 and not as many ring structures. So to wrap up, hopefully I've convinced you today that the SMILE sequence and molecular graph structures can contain complementary information, and that we can integrate these at the treatment level. And we think this helps Pisces better navigate some of the trade-offs, like the number of rings and the molecular graph diameter, particularly when our basic rule of thumb might suggest that we should make different modality decisions for each of the two drugs in the combination. In the future, we'd like to incorporate chemical language models into Pisces to encode the sequential drug inputs like the SMILE sequence, both to better leverage contextual information in the representation, as well as the power of large language models. Although today I focused on drug synergy prediction, we'd also like to apply Pisces to more drug combination tasks, including drug-drug interaction to predict things like side effects. And finally, we think we can do a better job at representing the cell lines by applying our contrastive learning framework for cell line embedding as well. So in what I've shown you today, we've been focused on the expression data for representing cell lines, but in the future, we'd like to include other modalities like mutation information to better represent cell lines with respect to treatment, and hopefully to further improve our performance on those challenging settings, like when we're applying the model to cell lines we've never seen before. So I'd like to thank all of my co-authors on Pisces at both Tsinghua and the University of Washington, so Jiacheng Lin, Hanwen Xu, Professor Jianzhu Ma, and Professor Wang, and thank you so much for your attention. I'd be happy to take any questions.